Hello student, today's topic is biosensor. As you see in these pictures, we will going through a kind of electronics or electrical devices, right? At present situation, most of our families are used to with these kinds of devices. And the obvious reasons of this COVID situations, we all are using oximeter, thermal detector, in our houses for our safety, right? So these kinds of devices like oximeter, thermal detectors, all are biosensor. In addition, the pregnancy test kit also a biosensor, right? According to our syllabus, we have to complete it the principle, working and applications of biosensor. So let's see what is biosensor. The technical definitions of biosensor is these are analytical device which measure concentration of an analyte. So here is a term analytes. So what is analyst? Analysts are the substance which are introduced into a biosensor for measurement or for analysis. Clear? Next points. In biosensor, biological materials interact with an analyte. So, after introducing the analyst within the biosensor, biological material interacts with the analytes. So, what is biological matters and where its place? So, biological materials or bioelements are those substances which are already present into the biosensor. And how it's work? It just interacts with the analytes and creates physical changes. As written in the third point, this interaction produced some detectable physical changes. After interactions in between the biological materials and analytes, it produces some physical changes, and these physical changes create a physical or electrical impulse or electrical signal, as you see in the third point, which is measured and converted into the electrical signal by a transducer. That means those physical changes are converted into an electrical signal by the help of a transducer. And next point, electrical signals is simplified and interpreted and displayed. So after creating an electrical signal, those signals are going through the amplifiers. After amplifying the electrical signals, it's detected by some software and then displayed the concentration of analyte. So here is a symmetrical diagram of biosensor. As you can see, we introduced analytes into the biosensor which react with the bioelement or biological materials. Then it creates some physical changes, and these changes with the help of transduction creates electrical signals which are introduced with amplifiers. An amplifier can detect the electrical signal properly, and those signals are going through the processor. And finally, it shows the results by display. And the most important point to be noted, Leland C. Clark introduced a oxygen detection biosensor called Clark electrode, and he is known as father of biosensor. And let's see an overview. Components of biosensor. First, analyst. What is analyst? These are the materials which are measured in biosensor like biological molecules, blood sample, vitamins, proteins, peptides. So, analyst is a substance which we need to measure, right? It may be molecules, biological molecules like glucose, fructose, or maybe blood samples, vitamins, proteins, hormones, any other substance. Next, number 2, biomolecules or biological molecules, right? These molecules react with the specific analytes and produce physical changes. So, what is biological molecules? Biological molecules are those molecules or substance, you may call it substance, which are already present into the biosensor and it reacts with the analyst, right? It may be enzymes, antibodies, polymers, etc. Next, the third number, transducer. What is transducer? These molecules generate electrical impulse like thermal impulse, magnetic impulse, electrical changes, electrical changes, optical changes, etc. So, what do you mean by transducer? Transducer is a substance or molecules which has to generate the electrical impulse from the physical changes. Physical changes means what? The reactant materials or reactant product in between the analytes and the biomolecules, right? Next, the display and the processor. Processor means some softwares which can detect the electrical impulse, right? And next, the last one is displayer. Displayer can display the result or the concentration of analytes. And the next, types of biosensor. What are the types of biosensor? So, first one is calorimetric biosensor. So, what is calorimetric biosensor? Release temperature or exothermic release temperature for exothermics and the absorb temperature or endothermic. That means which type of biosensors, which principles is depends on the release of temperature, that means exothermics, and the absorb of temperature, that is endothermics. That means those biosensors which principles is dependent with the temperature release or temperature absorb, we may call this biosensors as a calorimetric biosensor, right? Next, potentiometric biosensor. The measure of potential difference arising by drug reaction. During drug reactions, it creates a potential difference, and those biosensors which follows these principles we are called potential biosensor. Next, amperometric biosensor. What is amperometric biosensor? They measure the current flow or flow of electron arising during reaction. That means those biosensors principles follows these principles are amperometric biosensor. Next, conductometric biosensor. The major changes in electrical conductivity during a reaction. The principles should be the major changes in electrical conductivity during reaction. In case of conductometric biosensor, it is the major principle. Next, number 5, acoustic wave biosensor, and the next word is acoustic wave biosensor. So, what is acoustic wave biosensor? The measure electrical field development by piezo or piezoelectric effect. What is piezoelectric effect? It is the ability to certain materials to generate an electrical changes in response to applied mechanical strength. Okay. So, examples of this kind of biosensor is cocaine biosensors, which have a popularity or you say the very much popularity in the sports field. And the last one is optical biosensor. In this biosensor, measure light arising from the reaction. This reaction involves with an enzymes, that is luciferase. We all know the luciferin, the light emitting compound found in the organisms or generated by biomolecules, which is present mainly in case of fireflies. In fireflies they have a light emitting compounds, that's called luciferins, and luciferase is related to luciferin, which creates a light. And next we will see type of physical changes in biosensor. Those kind of physical changes which can create or by which electrical signals are created. So what are the types? First types is heat release or absorbed by reaction. As you see in the calorimetric biosensor, there is a principle of absorption and release of heats, which is related to heat release or absorbed by reaction type of physical changes. And the next one is potentiometric biosensor. In potentiometric biosensor, there is a physical changes, that is electrical potential due to change in electrical distribution or electron distribution, right? And the next one is light produce or absorb. In case of optical biosensor, we can see the measure of light arising from the reaction, kind of this, light produce or absorb. Next, the movement of electron due to the redox reaction. Redox reactions means ki redox is related to oxidation and reduction reaction. That means some kind of biomolecules or bioelements and analytes going through this redox reaction and create electrical impulse by moving electrons. And the next one is mass changes in biological components. That means in some biosensor, analytes and the bioelement react and create a change in biomass. And the next, the use of biosensor or uses of biosensor. Biosensors have been used in different fields. Some of these are medical diagnosis, study biomolecules, food analysis, drug adulteration analysis, industrial effluent controls, pollution controls, virus and bacteria analysis. In medical diagnosis, some detection of glucose, detection of urea, detection of any kind of disease, viruses, it's included in medical diagnosis, right? Study of biomolecules. Biomolecules means some kinds of cellular materials, like some kind of like carbohydrate, lipid, nucleic acid, that means cellular materials, right? In case of food analysis, in food analysis we can analyze or detect the purity of the foods and adulterations, how much adulterations have in these foods, we can analyze this. And drug adulteration also can analyze by a biosensor. In case of industrial effluent controls, the effluents means, what is effluents means? Effluents means liquids, waste or swags discharged into the rivers and the sea. So industrial effluents means those effluents which are created by industries. So biosensor can help to detect the industrial effluents, and we can easily controlled by this biosensor detectors. We can easily control this industrial effluents by this biosensor, right? And the pollution controls. So with the help of the biosensor, we can detect some unhygienic materials or gases like methane, CO2, etc. And for the virus and bacterial analysis, as we